morning. So the title of my paper is Targeting Human Cancer by Glycosaminoglycan Binding Malaria Protein, or in simpler terms, using a protein from malaria as a cancer-specific targeter. So first, I'm going to talk about current cancer therapies, and more specifically, chemotherapy. Chemotherapies target the entire area around tumors, hurting both the healthy and cancer cells, and this can cause side effects such as loss of energy, nausea, fatigue, and even more harmful side effects. So the researchers of this paper were looking into finding a treatment that would only target the cancer cells, avoiding the healthy cells, and hopefully avoiding most of the side effects. So the researchers asked themselves, what if there was a way to use the powers of malaria, a harmful parasite, to treat cancer with minimal side effects? So first, they wanted to find a cell in the body that's similar to cancer cells, so they looked at placental cells, because both placental cells and cancer cells have high rates of proliferation and they invade other cells for nutrients. And specifically on the cancer cells, they looked at the placental-like CS or PLCS receptors. Next, they looked at the malarial protein RVAR2. And from previous malarial studies, they know that RVAR2 binds to the PLCS, the receptors on the placenta. So first, the researchers looked at whether PLCS is apparent in only placental cells or in other healthy cells in the body. So they introduced RVAR2 into tonsil cells and placental cells, and as you can see, the brown is showing the RVAR2 binding to the PLCS receptors in the placental cells, but not in the tonsil cells. So the researchers did this throughout a whole group of healthy cells, and they saw that only in the placental cells was there RVAR2 binding, not in any of the other healthy cells. So this means that there's only PLCS in the placental cells. So next, the researchers looked at whether PLCS, the receptor, is present in cancer cells. So they hypothesized that it would be, and they were correct. They found that the PLCS is present in 95% of all cancer cells, except for this one cancer, Ewing sarcoma. And here you can see the red arrows are pointing to the PLCS on the surface of 3 different cancer cells, but not on the healthy cells. So why do you think it's important that there's PLCS on cancer cells? And why do you think the researchers thought it would be there?
So the PLCS is...
Wait. Can you ask somebody other than him? Okay.
No.
Okay.
So the mFAR2 binds to the PLCS, right?
Yeah.
So if they know that the PLCS is in cancer cells, in theory the mFAR2 will also bind to that PLCS that's in the cancer cells. So maybe they can use the mFAR2 in cancer cells.
Yeah. Okay.
Yeah.
So next, the researchers looked into RVAR2 tumor binding, so they know that there's PLCS on both the placental cells and cancer cells, and they know that the RVAR2 binds to the PLCS on the placental cells, but they need to confirm that the RVAR2 will bind to the PLCS on the cancer cells. So they took a stained stage 2 cancerous breast tissue here, and they introduced RVAR2 with CSA, and RVAR2 with CHase. And CSA are just receptors that are not on the cell, and CHase is an enzyme that removes the receptors from the cell wall. So as you can see, there's only RVAR2 binding when RVAR2 is by itself. When there's CSA or CHase, it looks just like the control. There's no binding. And here you can see in 3 different cancer cell lines, only with RVAR2 is there binding to PLCS. When there's RVAR2 and CSA in all 3, it looks almost like the control where there's no binding. So next, the researchers,
So is CHase made by the body, or is it?
Yeah.
Or is it? Okay.
Yeah.
Does CSA remove the PLCS receptors from the cell, or does it just add other receptors?
It's just other receptors like that aren't attached.
So that adds other receptors, and the other one takes off the original receptor.
Yeah, the CHase removes the receptors from the cell wall, so there won't be any receptors. Yeah.
So is CSA like a competitor to PLCS, or like
not really. It's just other like receptors, like kind of like floating around. So it's just like testing another type of receptor. So next, the researchers looked into whether the RVAR2 will bind to the surrounding healthy cells or only to the healthy or only to the cancer cells. So they took a whole group of cancer cells and their surrounding cells, but this is just a sample. And so these are the cancer cells and these are the natural adjacent tissues. And they found that only in the cancer cells was there RVAR2 binding, not in the healthy cells surrounding them. So why is it important that RVAR2 only binds to the cancer cells and not to the surrounding healthy cells?
It can be specifically to target cancer without hurting the cells too.
Yep. So next, the researchers looked into if RVAR2 could be an effective drug carrier. So they combined RVAR2 with DT, or diphtheria toxin, which is a known toxin to cells. And as you can see here, these pictures are showing RVAR2 surrounding to, binding and infiltrating a cell, a cancer cell. So the RVAR2 is in green, and the cancer cell is in blue. So it's surrounding the cell and then binding to it. And then here you can see where the cell has turned green, so it shows the RVAR2 infiltrating the cell. So this means when the RVAR2 is combined with the DT, that it will carry the toxin throughout this entire process, and when it infiltrates the cell, the toxin will also infiltrate the cell, destroying the cancer cell. So finally, the researchers looked into the long-term effectiveness of the RVAR2 DT treatment. So here you can see the black line is showing healthy cells, and these three colored lines are showing cancer cells. So this graph is showing the survival rate of the cells after RVAR2 DT is administered. So almost all of the healthy cells survive, while almost all of the cancer cells die from the RVAR2 DT. Next, the researchers took two groups of mice. One they gave saline, and one they gave the RVAR2 DT. And 10 days after the treatment, they found that the mice that received saline, their tumors had grown exponentially, while the mice that received RVAR2 DT, their tumors had stayed the same. They hadn't grown. So they concluded that for 10 days after treatment, the tumor stopped growing. Then they concluded that this drug only needs to be administered once every 10 days. But this is, even though it may be a little more frequent than chemotherapies, for example, you're avoiding most of the side effects. So in the time between your treatments, you won't have all these harmful side effects. You'll be able to be active. So a question for the researchers to consider is, what if the patient is pregnant and they have a placenta? So they know that in non-pregnant mice, there's no placenta, so there's nothing else for the RVAR2 to bind to. But what if the mouse is pregnant and has cancer? Would the RVAR2 DT bind to the cancer cells? Would they bind to the placenta? Would they bind to both? Would they only bind to one? So this is something the researchers need to look into. So overall, the researchers found that the PLCS, the placental receptors, are apparent in both the placental cells and the cancer cells, and that the RVAR2, the malarial protein that acts as a vehicle, binds to the PLCS on both the placental cells and the cancer cells. So as you can see here, the pink is showing the cancer cells, and the white is showing the healthy cells. So the blue is the RVAR2, the red is the DT, and this is just like a fluorescent molecule. But the RVAR2 is only binding to the cancer cells, not to the healthy cells. And when combined with the diphtheria toxin, or DT, RVAR2 can target the cancer cells while carrying the toxin, and it won't affect the surrounding healthy environment, since there's no PLCS and the RVAR2 won't bind to the healthy cells. And finally, the researchers concluded that the treatment lasts for 10 days before a new dose needs to be administered. So overall, RVAR2 has the potential to be a much more effective treatment for cancer, because it has a similar efficacy to chemotherapy, but you're avoiding most of the side effects. it's currently in clinical trial stages, but what this paper is showing is that the powers of harmful parasites like malaria that hurt so many people worldwide can be their powers can be harnessed and used for medical treatments that will help so many people in the long run. So thank you to all these people. Without them, this wouldn't be
Did any of the cancer shells begin showing any resistance to the treatment at all, or?
They didn't really cover that in the
Okay.
paper.
How did they determine it was only good for 10 days? Like, how did they determine 10 days was there?
Well, they saw that the tumors stopped growing for these 10 days, and then afterwards the tumors would start growing again.
Okay.
So they didn't give a second dose, and then the tumors started growing again.
So if they saw that the tumor only like stopped growing for 10 days, would 10 days only maintain the size of the tumor, or would it like shrink it?
Well, this, as you can see, it's all the tumors. They pretty much stay the same size, so that I think it's maintaining the size of the tumor, so it's preventing it from growing.
What is the drug toxin? Like, is it a specific drug?
Well, it's just diphtheria toxin, so it's like a toxin to all cells. So when it's
Okay, so
Did the paper go into why the good sarcomas were different?
not really.
All right, next week, Alyssa.
Unless you chat with something on OCD. Yeah.